Well, hey guys, this is Dr. McDonald. We're going to start on, see, this is part 3. I think we'll probably wind up with 4 parts. So let's talk about this. This is so important because we use this everywhere. This is how we chain together molecules to make bigger molecules. This works with our sugars and with our amino acids and with our nucleotides and with our triglycerides. I mean, it's used everywhere. So now that you know the importance of this, you got to keep it straight too, because people confuse these two, dehydration synthesis and hydrolysis. Synthesis means to put together, so that's the key thing. Lysis means to break apart, so that should make it a little bit easier to keep it. I'm just telling you what I've seen over the years as far as the testing. I've seen folks, they just get it exactly backwards. Okay, enough of commercial. Okay, so we have 2 monomers. A monomer is just a simple unit that when you chain them together, they make a polymer. A good example of that is starch. Starch in plants is a lot of glucose molecules that are chained together. It's just a way to store them. It makes it more convenient. So how do we do this? Well, we need an enzyme. We need something to help catalyze the reaction, and we'll talk more about this in just a minute. But if you notice on these 2 monomers, there's a OH. Those are called hydroxyl groups. There's one there and there's one here. Now, if I put the right kind of enzyme, I will break off that hydroxyl group from here, and on this second one, I'll break off just a hydrogen, not the entire group. Well, that's 2 hydrogens, excuse me, and 1 oxygen. That's water, guys. So what happens is I remove the water. That's why it's called dehydration. What am I left with? I'm left with an open bond right there, an oxygen that has an open bond. So all I really need to do is get that oxygen to form a bond with that first monomer right there, and that's it. That's it. You got it. That's just as simple as that. So by removing the water, the 1 oxygen that's left is going to form a bond. Now, in amino acids, it's not actually a hydroxyl, but I'll show you that when we get to the amino acids. So that's dehydration synthesis. Hydrolysis. So we in people and animals, we don't make starch, we make glycogen. It's very similar to starch, a little bit more branched. But we make that, and so in the middle of the night, when it's been a long time since you've had any food, your blood sugar level starts to decrease. Glucagon will be secreted from your pancreas, and that will cause you to go to the liver and break apart some of that stored glycogen. Now, how do we do that? It's just the exact opposite of what we did to chain them together. So if you look right there, there's that. That's not really a polymer, it's just 2, but we'll just keep our terminology consistent. There's a polymer, there's that oxygen bonded to the 2 monomers. So what I do in this case is I add water and breaks it back up and puts it exactly where it was. Instead of having that oxygen bonding these 2 monomers, I have a hydroxyl group here and a hydroxyl group here. Hydrolysis, breaking apart with water. Now these things, I mean, they have to have enzymes, guys. They don't spontaneously do this, but this is how we chain things together. You need to know this because it's going to apply for a lot of biochemistry in A&P1 and in A&P2. If you look down there at the very bottom, you'll see we've got an example. There's glucose and fructose. Those are single sugars. We'll talk more about them in just a minute too. But what we want to see is that there's a hydroxyl group there and a hydroxyl group there. If I take that hydroxyl, that OH off, add that one hydrogen, again, I've got H2O. I'm taking the water away. I'm dehydrating it. I've got that one oxygen left, and it forms that bond right there. And glucose and fructose happens to make sucrose, which is table sugar, which you put on your cornflakes. Okay, and then I can reverse this. I eat my cornflakes with the sugar on it. I get this in there. I cannot absorb sucrose through my GI tract. I have to break it apart into its individual sugars. How am I going to do that? Hydrolysis. I add the water and that special enzyme, and it breaks it back and puts it back where it was, and then I can absorb those. Just an aside, lactose, which is milk sugar, is a disaccharide. It's two simple sugar molecules bound together. People who are lactose intolerant don't have the enzyme to break it. They can't do the hydrolysis, and it's not absorbed in the GI tract, so it has to it just has to pass through. It gets to the colon, where it causes lots and lots of trouble. The bacteria in there will go crazy over that, and there'll be bloating and pain and gas and so forth, diarrhea. But in any case, I just thought that little clinical correlation might be interesting. Okay, let's go to the next slide. Let me move my picture. Okay, organic compounds, carbon, not carbon dioxide or carbon monoxide. And we've already said it's got to be carbon and hydrogen. Unique to living systems? No, not really. You can have organic molecules that are not related to living systems. So that one's not actually correct. The four main categories are carbohydrates, carbs, lipids, which are fats, proteins, and nucleic acid. Most of those are going to be polymers. They're going to be chains of similar units. The best way to think about monomer and polymer is monomers are the basic building blocks. They're like a Lego. You take a bunch of Legos, put them together, and you make something, a polymer. And again, dehydration, synthesis, and hydrolysis is the way that we do it. It's pretty straightforward. Carbon chemistry, we talked a little bit about this, that carbon has 4 electrons in its outer shell. Having 4 electrons means that it needs to form 4 bonds. It's not going to get an ionic bond because otherwise it has to either get 4 or give away 4, and that's not realistic. So carbon is one that forms the covalent bonds. Now there's the methane. You see a carbon, and each single bond is with a hydrogen. It just needs to share one pair of electrons. Now here's one where it only has 2 hydrogens. Now that has a single bond, and it has a triple bond to another carbon. That's not quite as stable as this, but you know, it works. Here we see one that has double bonds between these carbons. So these guys have 2 hydrogens, and he double bonds with this carbon. So he has 2, he has 1 hydrogen, and then he single bonds to another. But we don't need to go into that kind of detail. Now here you see a ring structure, and there's a lot of ring structures with carbon atoms. We'll talk more about that later. Here's a little bit more complicated structure. You can see that carbon is just a really good atom to form long chains of molecules. And so, as far as the functional groups on organic compounds, we've talked about the hydroxyl polar because that hydrogen can be released. And you find it just on every type of organic compound. Carboxyl groups mostly are going to be the fatty acids and the amino acids, and this is the part that makes them an acid. That's a carbon, 2 oxygens, and a hydrogen. So that carbon forms a double bond with that one oxygen and then a single bond with this OH group, that hydroxyl group. And then of course this bond is to whatever molecule. But you see that, and I'll show you that when we get there. Down here you can see the hydroxyl and the carboxyl. The amino group, these two are actually both found in amino acids, is an NH2, and you can see it right there. The methyl group, you remember methane was CH4. If instead of having 4 hydrogens, I have 3 and I bond to something else, we call that methyl. And then the phosphate group, this guy is charged. He has got a -2 charge. In other words, he's got 2 extra electrons. And he is very polar. We're going to find him in the nucleotides. We're also going to find them in the cell membranes, but you know, we'll get to that. Carbs, lipids, proteins, and nucleic acid. These guys are large. They have these functional groups. The functional groups can cause reactions. We've already talked about the definition of monomer and polymer. We'll keep going. Okay, carbs, sugars, and starches. Now here's an interesting little factoid. Carbon, hydrogen, and oxygen make up these sugars. And however many carbons you have, you're going to have twice as many hydrogens, and you're going to have the same number of oxygen. Good example is glucose is C6, and so I would expect the H to be 12, 2 times 6, and the O would be the same as the carbon, and it'll be 6. So C6H12O6. So these guys, we talk about the monosaccharides, which is the kind that we can actually take into our GI tract. The disaccharides, we talked about sucrose, we'll talk about some more. And then the polysaccharides, which are the long chain, the glycogen and the starches. The main fuel that we run on is glucose. We also use some of these sugars as part of our DNA and RNA. They make their structural molecules, and I'll show you that too. Okay, so the simple sugars have anywhere from 3 to 7 carbon atoms. Here's some of the most, there's the glucose. That's our normal blood sugar. 6-sided. One of the carbons is actually outside the ring, and there's oxygen in there, but it's a ring structure, 6-sided. They call it hexose. Now, fructose is, by the way, this guy has the exact same formula, C6H12O6, but they're rearranged. 2 of the carbons are off of the ring. It's still a hexose sugar. Fructose, is this the kind of stuff you get in fruit? Yep, that's it. And then we got galactose, which is another hexose sugars. These are all simple sugars. Now the pentose sugars, the 5-sided deoxyribose and ribose, they make up RNA and deoxy-DNA. If you notice, the only real difference between them is if you look right there on this corner, there's a hydrogen, whereas over here it's a hydroxyl. That's the difference between deoxyribose and ribose, not very much. Here are our disaccharides. We talked about this. Glucose bound to fructose is sucrose. If you get 2 glucose bound together, that's maltose, as in malted milkshakes. And here's the lactose, which is galactose and glucose. This is the one that not everybody has the enzyme to break it down. And so if it's not broken down into simple sugars, it does not get absorbed. And it's going to cause all kinds of GI upset down the road. Now the polysaccharides, now they're showing you glycogen here. Again, it's just a long chain of glucose. Now glycogen branches more than starch. Starch is the way plants store these. They branch, but they don't branch as much. Why do we branch so much? Because we have to break these off through hydrolysis one at a time. The more branches, the more you can break off at the same time, and the faster you get it out. And that's what we do in the middle of the night when our blood sugar runs low. Okay, proteins. peptide bonds are what we call these things. These guys are going to be, we're going to use the same dehydration synthesis. The only thing different is, if you look over here, you see this OH right there and this H right there. We take those out as water, and that carbon and that nitrogen are bound together. It's a dipeptide bond. The amino acids have carbon, hydrogen, oxygen, and they got to have nitrogen because it's an amine group, right? Sometimes you'll have phosphorus and sometimes you'll have sulfur. So this is, in general, this is what it looks like. If you look at it, you got a carbon with a hydrogen. Attached to one bond is the amino group. That's what makes it amino. Attached to the other side of that carbon is the COOH, the carboxyl group, which makes it an acid. Up here, this little green box is just an R. What does that R mean? That R means that there are different things that go in that box. There's like 20 amino acids, and the differences between the amino acids have to do with what is attached up here. The simplest amino acid is glycine, and that R group is nothing but a hydrogen. Aspartate, or aspartic acid, has a CH2 and then a COOH. Lysine has this 4-carbon chain that has an amine group. And cysteine, if you notice, this actually has sulfur in it. So these amino acids are going to be bound together to make our proteins. We're coming up on that. So a dipeptide is just 2 amino acids. Tripeptides are 3. Now, we can absorb 2 or 3 amino acids. They don't have to be completely broken down. We will eventually break them down into singles. Why are we doing that? Because I'm going to take this protein, cut all those amino acids up, and then I'm going to use those amino acids to build my own proteins. The collagen and the structural things that hold my body together, as well as the enzymes that run our chemistry. You are what you eat. Yeah, really, you are. And then, of course, we have the polypeptides, which are long. These aren't branched, and we're going to show something about that coming up. I don't want to jump the gun. A protein is a polypeptide that has changed shape. It's folded, and I'll tell you all about that coming up, guys. But anyway, you can see, like I was talking about, there's the OH from the carboxyl, one of the hydrogens for the amino. We remove that with dehydration synthesis. That nitrogen and that carbon bond together, and you can see it right there. And that's how we bond all these guys together to make our proteins. How do we break them apart? Hydrolysis. Same thing, guys. Same exact thing. Okay. We're going to talk about the structures of amino acids that make up these polypeptides and proteins. And I can't remember, guys. I think a polypeptide is anything less than 50 amino, and anything larger is a protein. It's just, I'm not even concerned, but you guys know that. Okay. Hold on. Well, we'll just keep going. Primary structure, if you notice, and you see these R groups on different sides, but you see there's the amine group. There's the carbon of the carboxyl, and you see it bound to the nitrogen. But the primary structure is just the sequence of amino acids. And which amino acid comes first and the next and all that makes different proteins. It's the sequence. That's the difference. Is it important? If you miss just one amino acid in your hemoglobin, you have sickle cell anemia. I think that's pretty important. So yeah, I like to think of it as if you had a string and you just put beads on it. Which color beads? I want a red here, green here, yellow, then I want a purple. Yeah, that's the primary structure of a polypeptide. Now the rest of it's going to be a little bit more mind-bending or polypeptide bending. Okay, I told you earlier that those hydrogen bonds are pretty weak, but they're responsible for a lot of molecular shape. Now if you look over there, that's the alpha helix. It's kind of a spiral shape. If you look right there between that red and that blue, those little dotted lines and the dotted lines here and here, what happens is different parts of this string are attracted to each other because of opposite charges, and they're going to change. They're going to take that one long bead on a string and they're going to twist it around. Now for secondary structure, they do one of two. They make this spiral an alpha helix, or they make this zigzag, which is a beta pleated sheet. They can have either one. Okay, let's do one more. Make it a little bit more difficult. Now we have the tertiary structure. I don't know if you guys can see, but that little spiral right there is an alpha helix. These little arrows, that's the way they represent beta pleated sheets. So in a single line of amino acids, once they do their alpha or their beta, then there's still going to be electrical attraction and it's going to bend even further. Now there are molecules called chaperones that help it to bend, but that's going to be the tertiary structure. Now that's as much as you can get with one string of amino acids, one protein or one polypeptide. That's it. That's as high as you can go with a single strand. We're not finished yet. So if I have more than one strand, if I have two, then that's going to be a quaternary structure. These whole things, this got primary, secondary, and tertiary, these guys are attracted to each other by electrical attraction, and they form a protein, but it's made of more than one string. As a matter of fact, probably the one that you're going to most hear about is going to be hemoglobin. Hemoglobin is 4 separate strings of polypeptides that are attracted. That's quaternary. This is, hold on a second, guys.